I'll talk to you about some of the work that we've been doing, and some of the speakers that you heard before are actually also involved in this, as well as Joan and Bob. And I'll focus on the growing pig. So just to give you a little bit of the history of how this research has evolved, at least the research on PRRS that I've been involved in. We started with experimental infections of nursery pigs with a single strain of the virus, and all these experiments were done at Kansas State. Done in total 10 trials with that virus, roughly about 200 pigs per trial. And some of those results have been published, or quite a few of those results have been published. And that was the start of the PRRS Host Genomics Consortium that Joan, Bob, myself, and Jim Reese started, with funding from USDA, NIFA, and the Pork Board, as well as the PHGC is actually a consortium of universities and quite a number of breeding companies that are operational in North America, so as well as some of the pharmaceutical companies. So very, very grateful for all their support. Okay, then we moved into additional trials that were funded by Genome Canada, actually the same project that John's pregnant gilt model 1 was funded by, as well as participation from the PigGen Canada companies, which again is a consortium of breeding companies that in this case have nucleus programs in Canada. So that was another 5 trials actually. And then we moved to perhaps a bit more realistic situation of co-infections. So we had experiments with co-infection as well as vaccination with a modified live virus, PRRS virus, and as well as field trials. And again, all that work was conducted at or by Bob's group at Kansas State, with funding from USDA and collaboration from Choice Genetics and PIC. And now a new project has started about a year ago with funding from Genome Canada, actually the same project that also funds John's second pregnant gilt model, where we are establishing or have established a natural challenge model, so with quite a number of pathogens present in that facility. And there's also funding from USDA NIFA that's part of that. Okay, so that first generation of trials where we infected the piglets with just a single strain of the virus, I think several of you have seen those results before. Using a GWAS, one of the SNPs or regions we identified was a region on chromosome 4 that had quite a major impact, a very significant impact, as indicated by the p-values that are on this graph, on PRRS viremia in serum over time. And with the AA pigs having higher viremia than AB pigs, the BB pigs, there are not that many of them. The B allele is not very frequent, and they have about the same effect as the AB pigs. So we are primarily focusing on the AA versus AB. The difference in viremia doesn't seem like very, very large, but it is on a log scale, and it is biologically quite significant, quite important, because it also was associated with quite a difference in growth rate, with the AB pigs having about 2.5 kilograms higher body weight or higher growth over 42 days compared to the AA pigs. Just presenting this in another way in terms of a bar graph, and now we're looking at viremia as area under the curve from 0 to 21 days. And again, if you focus on the AA pigs versus the AB pigs, so the darker orange and the light orange bars, you see a significant difference with the AB pigs having lower viral load, as we call it. When you look at weight gain, the AA pigs are again unfavorable. They have lower weight gain. And then this was repeated with several trials with a KS06 strain of the virus, which is a a strain that is less virulent, less pathogenic, and you can already see that with overall the viremia levels being lower than for the NVSL strain. But again, pigs in that category. However, when we looked at weight gain on the right side of the graph, there was no significant difference in weight gain, although there is a numerical difference, and this probably reflects the lower pathogenicity of this virus, where we do see an effect on viremia, but biologically, in terms of weight gain, it's not affected. There is some additional work that's been done there, and actually Laura Constance has a poster here that looks at differences in mortality and morbidity between those two strains of the virus. We have done additional work, and we actually were able to identify what we think is the causative mutation in this region on chromosome 4, and it's the guanylate binding protein 5. By the way, this is a region that you didn't see in John's presentation, so it's a region that was not significant for reproductive PRRS, but it is significant for the grow-finish animal. Interestingly, in the recent, actually the last couple of months, there's been work that has been published from the human field, where they find that this same gene, GBP5, also is associated with HIV, and in fact that they've identified GBP5 as a novel restriction factor of HIV that reduces the infectivity of the progeny virions by interfering with the processing of the viral envelope glycoprotein. And you can see that in the line graph there, that as the expression of GBP5 increases, the infectivity of the virus that is produced goes down. And they actually did a truncation of this gene, or they did an early stop codon that they knocked part of the gene out and producing a truncated protein, roughly at the same point where our causative mutation has an alternative splicing variant that also results in an early stop codon. And they also found that this truncated protein was not functional and did not reduce infectiousness of the virus. So it's a very similar effect to what we see, at least in terms of the viremia for the effect of GBP5 on PRRS. So now the next set of trials were these vaccination and co-infection trials, where we, and actually Megan already showed some results from one of those trials. Again, they were conducted at Kansas State, where they were 200 pigs again came to Kansas State. They were put into one or two rooms. One of the rooms was vaccinated with a modified live virus for PRRS. And then both rooms, all the pigs were, 28 days later, they were co-infected with PRRS and PCV2. And this was funded by USDA NIFA. And I'm not going to show you all the detailed results, but we did find that this chromosome 4 region, GBP5, again had an impact on the viremia for PRRS, both at the vaccine and in some cases also after co-infection. But it was quite interesting when we looked at PCV2 viremia after co-infection. There was also an association of GBP5 with PCV2 viremia, not in pigs that were not vaccinated, and that's shown here. So the non-vaccinated AA pigs being the white line, and the non-vaccinated AB pigs being the red line. So no effect of GBP5 on PCV2B viremia without vaccination, and that's consistent with all the studies that have been done at Nebraska by Daniel Chibano and others, where they infect with PCV2. They don't find GBP5 to have an effect on PCV2 viremia. But when we looked at the vaccinated pigs, so the pigs that were previously 20 days earlier were vaccinated with a modified live virus for PRRS, well, first of all, we saw an increase in PCV2 viremia, as shown by the solid lines, and we also saw a significant higher PCV2 viremia of the AA pigs versus the AB pigs. Okay, and so remember, AA was unfavorable in terms of PRRS viremia, and here we see it's also unfavorable for PCV2 viremia if the pigs were first exposed to PRRS through a modified live virus. And that's going back to a question that was asked this morning, what's the effect of vaccination with a modified live PRRS virus on other pathogens, response to other pathogens? I think this points out that we have to look at that. That's an important question we have to look at, because these viruses, they interact with each other, and PRRS makes cells more permissible for PCV2. And therefore, if you have an AB pigs that is a bit more resistant to PRRS, it doesn't, the vaccination doesn't have as large of a negative effect on PCV2 versus a pig that is AA for this region. Another question is, and that's a question that the industry, the breeding industry, has asked us, is, well, yes, good enough. You can select. You can tell us that we have to select for the B allele in this region because it gives some protection to PRRS when they're infected with PRRS. But what happens if they're not infected, when they're in a clean environment? And the other argument there is that the B allele, the favorable allele, has low frequency. So why is it so low? Is there some negative effects? And so one of my graduate students who is here, Jenelle Dunkelberger, spent the summer in the Netherlands working with Pramwood Mathur, who is here also at Topics Noordsvin, and analyzing the large database that I have of lots of traits on pigs collected on multiple lines in a clean environment. And she has a poster here that has all of the details, but the bottom line was that when they looked at the overall selection index value, that the GBP5 QTL had no effect, no significant effect on overall genetic merit across traits. And so that has given them encouragement, and actually Topics Noordsvin has now made the decision that they will start selecting their lines, at least some of their lines, for the B allele for this region. So as a conclusion, and before I go on, this is not quite the end yet, so marker-assisted selection based on the GBP5 genotype is promising, not only from the standpoint of PRRS, but perhaps also with regard to other pathogens in terms of co-infections. And the second point there relates to the effect on PCV2. And also, Jenelle has done work comparing primary exposure versus secondary exposure, and numerically the effect of GBP5 was greater upon primary exposure, either as a vaccine or as the actual virus, and that's consistent with the role of GBP5 in innate response. So next I move on to transcriptome analyses, and I don't have the nice summary results that John had. We're not quite at that point yet, but we have done quite a bit of work, or the collaborative team involved has done a lot of collaborative work, and a lot of this part has been led by Joan Lunny, initially looking at samples from these trials, picking high responders versus low responders, using microarrays, and then subsequently also using RNA-seq as shown when comparing AA pigs versus AB pigs. We are doing additional transcriptome analyses. Jesse Dong, another graduate student in my group who is here, together with Joan and others, we're doing two types of transcriptome analyses. One is to look at these co-infection trials and try to identify genes that are differentially expressed as a result of either vaccination and co-infection. And then also looking at the tonsil, looking at gene expression in the tonsil as a reservoir for persistent PRRS. So that first trial is still in the works, but we have done some of the initial assays. So what we did is we went to one of these co-infection trials, actually I think the same one that Megan used, where we have non-vaccinated pigs and vaccinated pigs, and we have AA pigs for this chromosome 4 region and AB pigs. And we found litters where they had one of each. So two that were vaccinated one of them being AA, one of them being AB, and the same for the non-vaccinated. And we found 7 of those litters. And then we, of course, we have samples. One of the beauty of these experiments is that they've collected lots of samples in Tempest tubes, and we were able to then get DNA. Jones' lab isolated the DNA, and then the RNA sequencing was done at Iowa State for a total of 192 samples. And one of the new things here is that we used what's called 3' RNA-seq, which is a new technology that only sequences the 3' end of RNA, of transcripts, and thereby substantially reducing the cost of doing RNA-seq work. A disadvantage is that you don't get alternative splicing information, but very cheaply it allows you to do large numbers of samples, and that's certainly a technology that we will be using extensively over the next little while. We're still in the process of further analyzing that data, so that's still to come. But the second one is tonsil gene expression, and actually Jesse Dong, who's here, has an abstract on this study, and so I'm just going to skip through this, also because of time. But you can go to her poster and find a lot of information about the relationship between serum viremia and viremia in tonsils, and the number of the different genes that she identified, and different networks or pathways that she identified. Finally, I want to talk about another big question in the industry, and that is, well, if we have the ability to select for improved health, not just PRRS, but in general, should we select for resistance, tolerance, or resilience? So just to explain those terms, resistance refers to the ability of an animal to either prevent infection or at least limit replication. So here I have 2 animals, A and B, and you have the level of exposure on the x-axis, and you can clearly see that animal A is more resistant. It still gets infected, but it doesn't, its pathogen load measured in serum or wherever isn't as high, doesn't increase as fast with level of exposure as the second animal. And so if we have, if they're both exposed to the same level, then the pathogen load that the A animal has will be less than what the B animal has, and so animal A is more resistant. Tolerance, which I realize is a loaded term in this audience, it doesn't have anything to do with vaccine effectiveness and so on. It refers to the ability of an animal to maintain performance as pathogen load increases. So pathogen load, again, measured as viremia, so that's on the x-axis. So as pathogen load increases, you can see that the decline of growth rate is faster for animal A than for animal B. So animal B is more tolerant to whatever damage the pathogen creates. Now, these 2 animals, they didn't have the same pathogen load. Animal A actually had a lower pathogen load to the same level of exposure. So that brings another term, which is resilience, which directly measures the relationship between level of exposure and growth rate. And so that's why when we talk about resilience or robustness, that's what we mean. And for these 2 animals, if you give them the same level of exposure, so if they're in the same barn, exposed to the same diseases, same level, there actually is not very big difference between, in terms of growth rate. It's actually animal B is a bit worse than animal A, but it will depend on the level of exposure that you have in the herd. So disentangling this, ultimately we're interested in animals that are more resilient, that also grow well when they are exposed to disease, but it may be important to disentangle tolerance and resistance, in particular because selecting for tolerance may put less of a pressure, selective pressure on the pathogen. So we can actually look at that in our data and using reaction norm models. Here what we've done is look at how the 42-day growth rate when pigs are infected with NVSL differs between AA pigs and AB pigs for GBP5. And you can see that the AB pigs have a slope that is not as steep as the AA pigs, and so the AB pigs are more tolerant. Now, this difference was not significant, but numerically there was a difference. Now, of course, the other thing that happened is that the AA pigs or the AB pigs were also more resistant because they had a lower viral load. Okay, so the AB pigs are more resistant, but they also tend to be more tolerant, and both of those are positive in terms of the difference we then see in terms of growth rate. So if we ignore the p-value and we call AB more tolerant, then the benefit of the AB genotype is both through increased resistance as well as through increased tolerance. For the KSO6 trials, though, it's a little bit different story. Still, numerically, we had less of a slope for the AA pigs than for the AB pigs. And there was also a difference in resistance, but in the end it resulted in a very small difference in growth rate. But you can of course see, it depends here where you are on the x-axis where that different, how big the difference in growth rate is. And so it's important to disentangle the effect of a particular gene or line on resistance versus tolerance. Now, to get at that, there's a new project that has started, funded by Genome Canada, and also will have funding from USDA, and again involving quite a large number of industry players through PigGen, and John Harding is one of the co-PIs on that, where a natural challenge has been established in a research facility in Quebec, in Canada. And the idea is that, again, industry will be providing commercial crossbred pigs, Yorkshire Landrace, and in a total, over a 2-year period, 3,500 pigs will be sent through this system. It's a continuous flow system that has been seeded with cedar pigs with a number of diseases, and I'll show you diseases that are present in a second. So the barn has been seeded, and then there's a continuous flow, so we'll have a continuous disease, substantial disease pressure in that line to express the genetic differences for resilience and robustness. They come into a quarantine nursery, and that also has an important role that I'll talk to you in a second. So these are the pathogens that were present as of October last year, and John Harding is very much involved in overseeing this, and as well as, of course, animal care, so that everything maintains a manageable levels in terms of the level of exposure and mortality. But you can see there's most of the major diseases have been brought into this barn. So these pigs are going to be exposed to quite a substantial challenge, and therefore we get very good expression of resilience. And that's what we're going to measure. So we are measuring, of course, growth rate, but we also get individual feed intake, we get individual water intake on all these pigs. Of course, mortality, morbidity, disease diagnostics, any treatments. Also, on a number of pigs, we get blood transcriptome work, so tempest tube, blood is collected in the tempest tube, as well as the gut microbiome. The quarantine nursery serves to be able to collect samples and data on young, healthy pigs. So these pigs come from they're weaned from clean multipliers and then brought to Quebec. And we're collecting lots of samples in the barn, immune response. Bonnie Millard is applying some of her high immune response tests from University of Guelph. And as well, John and others are involved in doing several in vitro immune tests. We're also taking samples to be able to look at the blood transcriptome, the blood metabolome, the blood proteome, and also the gut microbiome, and Megan will be interested in this. And again, this is done at a clean phase at a young age. Yeah. Couple of the data, and then I'll show you a little bit more of what we want to get out of this. So this shows the individual growth data. So they are weighed on a weekly basis, and this represents 7 batches of about 60 to 75 animals. So they come in every 3 weeks. A batch of 60 to 75 pigs comes in from a different source. from 7 different companies, breeding companies. So a lot of variability. And this is from one batch, the individual feed intake that we're getting in the finisher. The blue ones are ones that died. And then the orange ones are ones that made it to market. We also get the slaughter data. But you can already see, for example, this animal 60, you can clearly see a decline in feed intake representing disease. And we'll be able to match that with water intake data, the treatment data, so to be able to identify on an individual animal basis episodes of disease. And then we have lots of diagnostics then also to be able to see what diseases were present in the herd at that particular point in time. And Austin Putz, graduate student in my group, will be analyzing a lot of that data. We also are going to get high-density SNP genotypes across the genome, so we'll be able to do GWAS and genomic prediction. And of course, with this in-depth data, lots of statistics and bioinformatics will be needed. Outputs are, on the one side, when we look at the resilience phenotypes that they're collecting in the disease environment, which are unique. As this detailed data is not available anywhere else on large animals that I know of. We'll be able to look at the genetic basis of these traits, parameters, architecture, and mechanisms. And then with the data we're collecting in the nursery, we'll be able to use those as predictors, predicting and either genetic predictors or phenotypic predictors. Idea would be to develop predictors that could be used in a nucleus farm that could be measured on young animals in a clean environment. So that's the lots of collaborators. The final thing that I'm going to leave the group with is as part of this USDA grant that we got. It's an integrated grant. It also has an important education component, and one of those is the development of an online course that is led by Monsi Toro-Morel out of Minnesota. Title is Principles and Applications of Genetics and Genomics to Improve Animal Health. And the target audience is veterinarians, so people like you, and geneticists, and to educate and update people on the concepts of genetic selection and genomics, and specifically their application to improve animal health. 4 units, disease biology, genetics and genetic selection, specifically applied to disease resistance, and also specifically applied to selection for improved resistance tolerance for PRRS.